Hello guys, how are you? Welcome to our channel Last Minute Pharma, your exam antidote. Today we are here with lecture number 7 of the general pharmacology series, and today we are here going to discuss about the various receptor families. Okay, so in the last lecture, that is nothing but lecture number 6, we have discussed about the receptor mediated and non-receptor mediated, right? The pharmacodynamic aspect of a drug, right? We understand about the receptor mediated mechanism, the non-receptor mediated mechanism, and various things like agonist we have discussed, what is antagonist we have discussed. You know, so today which we are there, we are going to mainly discuss about the various receptor families. It's a topic of a receptor mediated type of transport itself. Okay, so before going through this receptor families, okay, you have to mainly understand these are the various receptors, okay, various types of receptors which are going to be present in our biological body. Okay, so any drug, okay, any drug which is there, when the drug is going to interfere, okay, when the drug is going to interfere in this receptor mechanism to show a therapeutic activity, right? That is what we have to understand with this particular receptors. Okay, again I will repeat. See, receptor mediated or receptor families these are different types of receptors which are there in the biological system. So now drugs which are there, okay, there are certain drugs which are going to interfere, okay, there are certain drugs which are going to interfere with these receptors, okay, with these receptors to show a therapeutic activity. So let's try to understand what are the different types of receptors which are available to us, okay. So the first one which is there, that is the ligand-gated ion channel, okay. We'll be discussing in detail about this, okay. What is ligand-gated ion channel, okay. This is also called as inotropic receptor. Next we are having the G-protein coupled receptor, and this is a 100% 5-mark question which will be coming in your examination for PharmD or B-Pharm syllabus, because this is 100%. Many times this question has come. I cannot guarantee about other receptors, but G-protein coupled receptor is one of the very important mechanism, and most of the drugs which are there are going to interfere with this G-protein coupled receptor only, or they are going to act on this G-protein coupled receptors as such. Okay, the next which one is there, that is your enzymatic receptors. Next one which is there, that is the enzymatic receptor. And then the last one which is there, that is the nuclear receptor, or receptor which is going to regulate the gene expression. Gene expression in the sense for transcription process. Okay, in the transcription process, these receptors are there. These particular receptors are there. So if the drug is going to interfere with this nuclear receptor, then what is going to happen? The gene transcription is going to get interfered. Okay, so we'll discuss in detail about this. Okay, we're going to detailedly discuss about this. Let us start discussing it one by one. Okay, so first one which we're going to discuss is about the ligand-gated ion channel. The first one which is there, we're going to discuss about the ligand-gated ion channel. So, ligand-gated ion channel, what does it mean? This is one of the examples. See, one example of the ligand-gated, if we talk about ligand-gated ion channel. Okay, so examples of ligand-gated ion channels are what? First, you are having your GABA receptors. Okay, you are having your GABA receptors. Then you are having your nicotinic receptors. Yes or no? Then you are having your nicotinic receptors. See, again I'm telling you, don't panic with the examples, because see, this is the first time, right? General pharmacology. You just have to focus on the mechanism of action. Once we start with the studying of the drugs, like ANS system we'll discuss, CNS we'll discuss, cardiovascular you will discuss. So once you understand that cardiovascular, ANS, then you will get a better form with this types of receptors. Okay. So now just relax and try to understand how the receptors are going to work. Okay, cool, fine. First is GABA receptors are there, nicotinic receptors are there, acetylcholine receptors are there. All these type of receptors which are there are going to work on the mechanism of the ligand-gated ion channel. Okay, so what does this ion channel mean? What does this ion channel means? Like various ions are there, right? For example, you are having your sodium, you are having potassium, then you are having your chlorine, right? Then you are having your calcium. These are the major ions. These are the major ions which are going to be there. Okay, now we will try to understand what is going to happen. Okay, so when the drug which is there, or we will call it as agonist, we will call this as agonist. When this agonist which is there, this is going to bind to the particular receptor. This is going to bind to the receptor. Now which receptor am I talking to? I am mainly talking about the ligand-gated receptor. I'm mainly going to talk about the ligand-gated receptor. So when this drug or when the agonist which is there, you know what is agonist, right? Which is going to have a higher affinity and intrinsic activity also is high. So when the drug is going to bind to the receptor, then what is going to happen? It is going to allow the flow of ions. It's going to allow the flow of ions. And once the ions are flown, what type of ions are there? I told you, right? You're having your sodium, you're having your potassium, you're having calcium, yes, you're having chlorine. So when the flow of ions are there, okay, when the flow of ions are there, what is happening? Receptor is there, agonist is going to bind, and when the agonist is going to bind, the channel, the ion channel which is there, either it will open or it will close. Okay, depending on the activity of the agonist, the ion channel might open or close, and this will lead to the flow of ions. Like it can be the sodium ions, potassium ion, calcium ion, or chlorine ion. And once the flow of ion is there, this will cause hyperpolarization. This will cause hyperpolarization, or it can also cause depolarization. It can either cause hyperpolarization or depolarization. Okay, and after showing this, once there is a hyperpolarization or depolarization, then what is going to happen? You are going to have the tissue response. Then you are going to have the tissue response. I hope it is clear. This is the mechanism of action of the ligand-gated channel. Okay, like for the drug which is there, agonist which is there, this is going to bind to the particular receptor, which is going to open or close the ion channel. And once the ion channel is open or closed, then the flow of ion is going to take place. And once the flow of ion is going to take place, then what is going to happen? It will cause either hyperpolarization or depolarization, and because of that, the tissue response is going to be seen. I hope the mechanism of action is clear. Okay, and one very interesting thing about ligand-gated channel is that these drugs or these receptors which are there, they show the activity or they show the action in milliseconds. This is one of the fastest mode. This is one of the fastest mode. The time taken for the drug, okay, for the drug which is going to bind to the ligand-gated, right? So for it to show the action, for it to show a therapeutic action, these drugs which are going to bind to the ligand-gated one are going to show quick action in milliseconds. Okay, see, I'm not talking about the bioelectric and all these things. Okay, I'm talking about the once the agonist is going to bind to a receptor, the mechanism of action, you say, or the speed which is there is comparatively fast. This is one of the fastest compared to other four: G protein coupled receptor, nuclear receptor, enzymatic receptor. This is one of the fastest mode. Okay, now let's come and try to understand this example. Okay, this is an example of a GABA receptor. Okay, the GABA is nothing but glutamate amino butyric acid. Okay, so not glutamic, that is a gamma amino butyric acid. Okay, this GABA receptor which is there, okay, this is mainly present in the neurons, okay, neural membrane which is there. Okay, so this GABA receptor, I'll just tell you, this is mainly going to have 5 units. See, again, I'm telling you, don't panic. Okay, don't panic, because I know, because when I started learning this general pharmacology lecture, okay, so usually what happens is these all are new terms to you, so starting it will be a bit difficult, but once you understand the CNS, ANS, it will be very smooth for you. Okay, don't worry, try to understand the MOA. Okay, so what is going to happen? There is some receptor, just called as GABA receptor, having 5 subunits. That's nothing but your 2 alpha is there, 2 beta, and 1 gamma. Okay, so now the drugs which are there, such as your benzodiazepine, okay, some drugs are something but an agonist now. This is a agonist. That is nothing but benzodiazepine. So this drug which is there, or an agonist, this is called as the agonist. This is going to bind with the GABA receptor. This is going to bind with the GABA receptor. And once this binds with the GABA receptor, what is going to happen? This channel which is there, the chlorine channel which is there, this is going to frequently open. This channel which is there, it is going to frequently open. Okay, and why is this frequently opening? Why is this frequently opening? Because benzodiazepine has been binded to this. Now, once this is frequently opening, what will happen? Chlorine ion will be able to pass. The flow of chlorine ion is going to take place. Flow of chlorine ions. And once the flow of chlorine ion is going to take place, then what is that going to result in? This flow of chlorine ion is going to result in hyperpolarization. This is going to result in hyperpolarization, and this is going to give you an CNS depression activity. This is going to show the CNS depression activity. I hope it is clear. I hope the ligand-gated channel is clear. What am I saying? This is one of the example. This is one of the example of ligand-gated ion channel. That is nothing but about the GABA receptor. So what is going to happen in the GABA receptor over here? this chlorine which is there, okay, chlorine ion which is going to be there, once the agonist is going to bind to the receptor, okay, once the agonist is going to bind to the receptor, the chlorine ion which is there is going to flow, and once the chlorine ion is going to flow, it will cause hyperpolarization and CNS depression activity. This is one of the example. So I hope it is clear how the ligand gated ion channel is going to work. Okay. Now we are going to discuss about the very important topic, that is G protein couple receptor. Okay, so now which we are there, we are going to discuss one of the very important topic. It is G protein couple receptor. Okay, this receptor is a 100% question for your examination. 5 mark question. I'm starting and telling you. Okay, last lecture also I told you this is a confirmed question for examination, sessional, internals, whatever it is. Okay, so if we talk about the G protein couple receptor, try to understand the mechanism. See, again I'm telling you, don't go with the drugs and everything now, okay, because you don't have, because it's initial, right, you don't have an idea about the drugs, you don't know about the classification of the drugs, so don't go on the basis of how the drug is going to act, how adrenaline is going to act, how acetylcholine is going to act. So you don't need to understand that, you have to understand the mechanism of this particular receptor, how the receptor is going to show the action. Okay, now let's try to understand about the G protein coupled receptor. Okay, so this one you can see, right, the zigzag one. See, here is the biological membrane. Okay, here you are going to have the cell membrane which is over here. Okay, just have a imaginary one. This is your cell membrane. Okay, so now in the cell membrane, if we talk about, this is called as your G protein coupled receptor. Okay, this is called as the receptor molecule. This is called as the receptor molecule, and this one which is there over here, this unit. Okay, this unit which is there over here, this unit which is there, this is called as your G protein. This is called as your G protein. So what does the G protein consist of? G protein consists of a G alpha GDP unit. It's going to have a gamma and a beta unit. So these 4 units which are there, this is going to form the G protein. Now try to understand, when this form, this G protein receptor, if we talk about, this is in an inactivated state. This is in inactivated state. So when we talk about inactivated state, that means there is no connection between this receptor. Okay, there is no connection between this receptor and the protein molecule. There is no connection between the protein molecule and the receptor. But now, as soon as the ligand comes and joins with the G protein coupled receptor, then what is going to happen? Then this protein which is there, okay, this ligand or this receptor which is there, this is going to go and bind this protein or this ligand, this which we can see, this is going to go and bind with the G protein. This is going to go and bind with the G protein. Okay, just try to understand over here. This is over here. Okay, as soon as the ligand is over here, as soon as the ligand is going to attach to the receptor, this is going to go and bind with the G protein. And as soon as it binds with the G protein, or as soon as it is going to get converted into activated state, what is going to happen over here? This GDP molecule which is here, this GDP molecule which is over here, this is going to get converted to GTP. And when the GTP molecule is being made, then what is going to happen? Dissociation of G protein is going to happen. What is going to happen? Dissociation of the G protein. So here G protein has been dissociated in 2 units. The G protein which is there, it has been dissociated into 2 subunits. One is the G alpha GTP unit, okay, and other one unit which is there, that is the gamma beta subunit. These are the 2 units which are going to be divided. One is the G alpha and GTP unit, and other one is the beta gamma unit. These are the two units which the G protein is going to get divided when the ligand is going to come, and then this GDP molecule is going to get converted to GTP molecule. I hope it is clear till here. Okay. Now next step, what is going to happen over here? This G alpha and GTP unit which is there, this is going to go and bind with the target enzymes and the ion channel, or the effectors you can say. Okay. These ones you can see that GS, GI, GQ. What are these called as? These are called as your effectors. These are called as your effector. Based on which ligand has activated the G protein, on that basis you are going to have different effectors. On that basis you are going to have different effectors. Okay, so now try to understand, this G alpha and GTP unit is going to go and bind with this effector. This unit which is there is going to go and bind with the effector, and whereas you can see beta gamma unit, right, this beta gamma unit is going to go and bind with certain enzyme, causing it enzymes or ion channel, which can lead to activation of some enzymes or the inactivation of certain enzymes. Is it clear? So mainly what I'm talking about the effector is GS, GI and GQ. These ones which are there, these are activated by the G alpha and GTP unit. This is going to be activated by the G alpha and the GTP unit. Is it clear over here till here? Okay, fine. Now next what is going to happen? Next what is going to happen once this G alpha and GTP is going to bind? There are three options which are there. Okay, three fate. It can either go activate the G stimulatory pathway, or it can activate the G inhibitory pathway, or it can activate the GQ pathway. Okay, let's try to understand that. Okay, don't worry, we'll make I'll just make a flowchart of it. We'll understand it way better. Okay, I'm just trying to make you an idea. I'm just trying to give you an idea how the mechanism is going to be there. Okay, now once this G alpha and GTP, if it is going to activate GS, what is GS? GS is nothing but stimulatory. Okay, GS is nothing but a stimulatory activity. Stimulatory. Okay, GS nothing starts for stimulatory. So this GS which is there, or G stimulatory which is there, G stimulatory protein which is there, or enzyme which is there, this is going to activate your adenyl cyclase enzyme. This is going to activate the adenyl cyclase enzyme. What is the function of this adenyl cyclase enzyme? This adenyl cyclase enzyme which is there, this is going to convert your ATP, this is going to convert your ATP into cyclic AMP. This is going to get converting this adenyl cyclase which is there. This is going to convert your ATP to cyclic AMP. And because of this, what is going to happen? The concentration of cyclic AMP is going to increase. And if the cyclic AMP concentration is increased, this cyclic AMP which is there, this is going to activate the protein kinase enzymes. These are going to activate the protein kinase enzyme. Okay, and activation of this protein kinase enzyme is going to lead to phosphorylation of the enzymes. Okay, this is going to lead to the phosphorylation to the enzyme. I hope it is clear. Okay, so what is going to happen if adenyl cyclase is activated, and when will the adenyl cyclase activity will be stimulated? When the G alpha and GTP molecule goes and activates the G stimulatory protein, then what is going to happen? Adenyl cyclase activity is going to be more. If adenyl cyclase activity is more, then what is going to happen? The cyclic AMP concentration is more. If cyclic AMP concentration is more, it is going to lead to the activation of the several protein kinase enzyme, which is going to lead to the phosphorylation of the proteins. I hope it is clear. Okay, fine. Now, next what is going to happen? This is one pathway. Exactly opposite pathway is going to happen with the GI. It is nothing but GI which is there. It stands for inhibitory. I which is over here, that stands for inhibitory. Okay, so what does inhibitory means? Exact the opposite thing. GS was stimulating adenyl cyclase, but whereas the G inhibitory which is there, this is going to suppress the adenyl cyclase enzyme, or this is going to act against the adenyl cyclase, going to block the adenyl cyclase enzyme. So if you are blocking the adenyl cyclase enzyme, then what is going to happen over here? Negative effect, that is decrease in the cyclic AMP level. This is going to lead to the decrease in the cyclic AMP level. That means what is going to happen? The enzymes won't be activated, right? Because no phosphorylation is happening. The protein kinase enzymes are not activated. Now, obviously, activity which is over here is going to be exactly opposite of the G stimulatory. For example, if G stimulatory is going to cause vasoconstriction, but the same enzyme, G inhibitory which is there, this will cause vasodilation. Okay, I hope it is clear. If you don't take the example, I'll tell you. Once we will go on the detail about the beta blockers, alpha blockers, and all these things, you'll clearly understand how these enzymes are going to work. Okay, now next which is there is the GQ pathway, okay, or the phospholipase C pathway. Okay, so what is going to happen in this phospholipase C pathway? This phospholipase C is an enzyme. Okay, phospholipase C is an enzyme. This is going to convert, okay, this is going to convert your phosphatidyl inositol 5-phosphate into two components. Okay, that is your DAG and IP3. It is your DAG and IP3. These two components or metabolites, this particular metabolite is going to get converted to DAG and IP3 in the presence of this phospholipase enzyme. Okay, and now what is going to activate the phospholipase enzyme, phospholipase C enzyme? That is your GQ. That is going to activate your GQ. Okay, so now once the DAG which is there, okay, this DAG which is there, this is going to cause activation of the protein kinase, and this is going to lead to the phosphorylation. This is going to lead to the phosphorylation. And if I talk about the IP3, IP3 which is there, this is going to bind with the endoplasmic reticulum. Where is it going to bind? Endoplasmic reticulum. And then this IP3 which is there is responsible for increasing the calcium levels. This is going to be responsible for calcium level, and if the calcium level is more, what is going to happen? Muscle contraction. Yes or no? So if the calcium level is more, what is going to happen? This is going to stimulate to the smooth muscle contraction, or you can just write over here muscle contraction. Okay, and that pathway which is there is going to activate various protein kinase enzyme and then going to activate the phosphorylation. Okay, see, this depends which enzyme is there which is going to activate. It all depends on what is the ligand going to come over here. Okay, based on what ligand, or the ligand means drug. Okay, based on the drug molecule or ligand molecule, it can take various pathways. One is your G stimulatory pathway, other one is G inhibitory pathway, other one is your GQ pathway. Then you are going to have one more, that's your gamma beta pathway. That means it's going to activate several enzymes or ion channels. Okay, so I know it might be confusing for you for the first time. Let's try to break it down. Okay, let's try to break it down for you in a simpler way. Try to understand this flow chart. Okay, so first what is going to happen? You are going to have a agonist. This agonist which is there, this is going to bind to G protein couple receptor. Okay, now once the binding has taken place, okay, once the ligand or the agonist, you can say, or I can say ligand, once the binding has taken place, what is going to happen over here? The GTP which is there, so GDP which is there, this is going to get converted to GTP, or this is nothing but you are getting activation. This process which is there, no? Once the binding is happening, this is nothing but the activation of the G protein. This is nothing but the activation of the G protein. Okay, so GDP has been converted to GTP. So what is going to happen over here? As I told you, your G protein couple receptor, or that G protein if you talk about, is going to have how many units? You're having alpha unit, you're having your GDP, then you're having your beta unit, and then you're going to have gamma unit. 4 units, right? But now this is a G protein. But now, once it is activated, what is going to happen? Dissociation of the G protein. What is going to happen? Dissociation of the G protein. And once the dissociation of the G protein has happened, then what is going to happen over here? This G protein is going to get divided into 2 subunits. This G protein which is there is going to get divided into subunit. That is your alpha GTP, because you see GDP is getting converted to GTP, so you're going to have alpha GTP unit, and other one is going to be your beta gamma unit. Okay, one is your alpha GTP unit, other one is your beta gamma unit. So if I talk about the beta gamma unit, it is going to either activate various ion channels or phosphorylate the enzyme. Okay, but now if I talk about the alpha GTP, okay, now if I talk about mainly the alpha GTP, try to understand. Okay, stay focused, because if you are not focused over here, you will miss it out. Okay, so if you are going to have alpha GTP over here, this alpha GTP can activate various effector organs. It can activate not organs, various effector enzymes. So what are various effector enzymes which I have discussed over here? That you are going to have G inhibitory, then you are going to have G stimulatory, and then you are going to have your GQ. So if you talk about G inhibitory and stimulatory, this is going to be your adenyl cyclase pathway. This is going to have your adenyl cyclase pathway. And if I talk about GQ, this is going to work on the basis of your phospholipase C pathway. Okay, so now I'm talking about G inhibitory. Let me talk about G inhibitory first. So what is this G inhibitory going to do? This is going to inhibit this adenyl cyclase. This is going to inhibit the adenyl cyclase, and because of that, what will happen? The conversion of ATP to cyclic AMP won't happen. So if the conversion won't happen, then what is going to happen over here? The concentration of cyclic AMP is going to decrease. So if the cyclic AMP is decreased, then what will happen? Will the protein kinase enzyme get activated? No. So will the phosphorylation of the enzyme won't happen? No. Okay. Now, if we talk about G stimulatory, it is going to activate. Okay, it is going to activate your adenyl cyclase enzyme. It is going to activate your adenyl cyclase enzyme. And once the adenyl cyclase enzyme is activated, what is going to happen? Exact opposite one, that is cyclic AMP. The concentration of cyclic AMP is going to get increased, then protein kinase activity will be increased, and because of that, this will lead to phosphorylation. This will lead to phosphorylation. I hope it is clear. I hope this part is clear. This is very important. Okay. Now, next one, if we talk about the phospholipase C, if I talk about the phospholipase C pathway, then what is going to happen? It's going to convert that phosphotinosyl biphosphate into your DAG and IP3. Okay. This is the DAG you are going to get converted and then into your IP3. This IP3 which is there, this is responsible for increasing the calcium level, and if the calcium levels are increased, this will lead to muscle contraction. This is going to lead to the muscle contraction. And if I talk about the DAG pathway over here, this is going to activate the protein kinase enzyme. And again this is going to lead to the same thing as their phosphorylation. So again the question comes, who is going to decide the fate of the particular drug? Okay, who is going to decide the fate? Whether the alpha ATP molecule which is there is going to, sorry, not ATP over here, just check it out, alpha GTP. Okay, so alpha GTP molecule which is there, either it is going to activate your G inhibitory or G stimulatory or GQ. It depends on the drug molecule which is over here. Okay, so I hope it is clear. Okay, this part is a very important part. G protein couple receptor notes are there in a Telegram channel. Okay, so you can actually go and check how you are supposed to write an examination. The proper diagram is there. Okay, so now we'll move to the third one. There is nothing but enzyme linked receptor mediated. Okay, let's see how the enzyme link is going to work. Okay, so now we are going to discuss about the transmembrane enzyme linked receptors, and the last one which is going to be there, that is nothing but the nuclear receptors. Okay, so I hope till now ligand gated channel receptors and then your G protein coupled receptors are clear. Okay, these two mechanisms are comparatively easier to understand. Okay, nuclear is very easy, but transmembrane again you have to focus to understand a bit. Okay, so see, what is a transmembrane enzyme linked receptor? I'll tell you in very short. Okay, see, these two which are there, they are polypeptide chains. Okay, these two proteins you can see over here, 1 and 2, these are nothing but the polypeptide chain, and this one which is there, the circle over here you can see, no, this is nothing but the tyrosine kinase. So now for example of an enzyme, it's an enzyme linked, right? Enzyme linked receptor. So the enzyme which is linked to this particular receptor is nothing but your tyrosine kinase. Okay, very simple, fine. Now, over here you have to understand, there are 2 units of tyrosine kinase. Okay, this which is there as such now, this is in an inactivated form. Now, how does this get activated? Whenever there is a ligand or a drug or a molecule which is going signaling molecules, okay, like insulin you can talk about, epidermal growth factor you can talk about, all these are nothing but the signaling molecules. Okay, so when the signaling molecules are going to go and join with this receptor, then what is going to happen? Once the binding takes place, this is going to lead to dimerization. Now, what is dimerization? Try to understand. These receptors which are there now, they are going to come closer. Okay, this is called as your dimerization. I'm just showing diagrammatic way. So this is your dimerization. Now they have been linked to each other. Now these tyrosine kinase has been linked to each other, and because of that, what is going to happen? This one which is there is going to be getting linked. So what is going to happen? This particular unit which is there, this has been converted into activated form. So when the signaling molecules were not there, when the signaling molecules were not there, it was in an inactivated form. But once the signaling molecules are came and bind to the receptors, signaling the receptors, then what is going to happen? They are going to come together, and that is what is called as the dimerization of the receptor. That's called as the dimerization of the receptor. Is it clear till here? The binding of the agonist will lead to dimerization of the receptor. And which one is this now? Which enzyme is linked to this particular receptor? The enzyme which is linked to this particular receptor, that is your tyrosine kinase receptor. Tyrosine kinase enzyme. Okay. Now, once this enzyme is getting activated, once the particular enzyme, this tyrosine kinase has been activated, what is the main function of this tyrosine kinase? Try to understand. Your ATP molecule which is there, with the help of this tyrosine kinase, this is going to get converted into ADP plus your phosphate molecule. And this one which is there, this is called as the stimulation of the intrinsic kinase activity. This step is called as the stimulation of the intrinsic kinase activity. Is it clear? Binding took place, dimerization took place, and because of dimerization, the activation of that particular enzyme, that is tyrosine kinase activity, started, and because of that, the intrinsic activity was seen, that was converting the ATP into ADP plus phosphate. Okay, now what happens over here? These phosphate molecules, see there are two molecules. No, here also tyrosine kinase is there, here also tyrosine kinase is there. So a lot of phosphate molecules will be generated. Okay, a lot of phosphate molecules will be generated. These phosphate molecules are going to bind to this particular enzyme. These phosphate molecules are going to bind with this particular enzyme, and this part is called as now, this receptor is phosphorylated. This receptor is here phosphorylated, or this is in an activated state. This is in the activated state. Okay, I hope it's clear. This has been phosphorylated and this has been in the activated state. Now, what is going to happen? See, after the phosphorylation, okay, intrinsic activity, what has been happened? Phosphorylation of the tyrosine residues. See, these all that circles you can see, the semicircles. These are nothing but the tyrosine residues. Now, after the intrinsic activity, they have been phosphorylated. Is it clear? Now, what is going to happen? There are various proteins. Okay, where there are various proteins which are there in the cell. And these proteins are going to go and bind with the phosphorylated kinase activity or this particular receptor. Okay. Now, for example, there is this protein. There is this protein A. This protein A will go and bind over here, and because of this binding, what is going to happen? There is going to be a cellular response. There is going to be a cellular response. But whereas imagine, for example, you are having protein B. Protein B. This protein B is going to activate or going to bind with this particular receptor, and then they are going to give you different. This is cellular response A, and here you can get cellular response B. So this is the mechanism of action, how this enzyme linked receptors are going to play. Okay, simple. I'll just revise it, try to understand. You're having two signaling molecules with a polypeptide with a tyrosine kinase activity, tyrosine kinase residues, but they are not phosphorylated or they are not in an activated state. So when the tyrosine or when the drug or like insulin which is there is going to go and bind to the signaling molecule, that is going to lead to the dimerization. What is that going to lead? That's going to lead to the dimerization. Now, once the dimerization has been taking place, then what is going to happen after dimerization? It's going to stimulate the intrinsic kinase activity. That means your tyrosine kinase which is there, it has been activated, and because of the activation of tyrosine kinase, your ATP is getting converted to ADP plus ADP plus phosphate, and this phosphate is going to phosphorylate this particular molecule. Okay, and now once this signal or this receptor is phosphorylated completely, there are various proteins which are going to bind to that particular phosphorylated protein or phosphorylated receptor, and because of that binding, you are going to have different types of tissue responses, you can say, or your other responses, cellular responses is what I'm going to say. Okay, this is the activity of your transmembrane enzyme linked receptor. Now, the last one which is there, very important one, the nuclear receptor. Very simple way. Okay, they work in a very simple way. See, till now we discussed about ligand gated, we discussed about your G protein coupled receptor, we discussed about transmembrane. All are going to be, or these receptors are all present where? They all are present in the membrane. But whereas if I talk about this nuclear receptors, if I talk about the nuclear receptor, and what does it mean? The receptors which are there, it can be either present in the nucleus or they can be present in the cytoplasm. Okay, either in the nucleus or in the cytoplasm. Now let's try to understand how the particular, this particular mechanism is done mainly for the drugs such as thyroxine, steroids like estrogen, testosterone, and then you are having your vitamin A and D. Okay, now try to understand over here that first this is your drug molecule, and inside or intracellular you are going to have the receptor. Okay, now what is going to happen? This particular drug is going to bind with the receptor, and this is called as receptor ligand, or you can say drug, or in the case of this mainly this steroid, so I can say receptor steroid complex. This is called as the receptor steroid complex, and once this receptor steroid complex is formed, this will be going inside the nucleus, and in nucleus what you are having? DNA, right? So this particular molecule which is there, or receptor steroid complex which is there, this is going to go and bind to the particular site of the DNA. This is going to bind to the specific site. Okay, this is going to bind to the specific site of the DNA, and by binding to the specific site of DNA, this is going to alter the protein synthesis. This is going to alter the protein synthesis. So you all know what is translation, right? Translation means what? You are going to convert the DNA, or you are going to convert DNA to mRNA, that is nothing but transcription, and from mRNA you are going to convert it to protein. But now, if this particular complex is going to bind to a specific site of DNA, then what am I going to do? I am going to alter the transcription. And if I alter the transcription, the mRNA will be altered, and if mRNA is altered, the protein which is supposed to be synthesized will be altered. So these drugs are going to work in that way. Okay, I hope that is clear. Okay, so this is going to alter the protein synthesis, and because of this, you can have the tissue response. Is it clear? So mainly the two types which are just now we discussed, that is your transmembrane enzyme linked receptor, and other one is your nuclear receptor. So now in the next, we'll just have a comparative study. Okay, we're just going to have the comparative study of the various kinds of receptor, their activity. Okay, let's go. Okay, so now we are going to discuss about the comparative study. Okay, so now we have finished all the four types of receptor families. There is nothing but your ligand gated, G-protein coupled receptor, enzymatic receptor, and nuclear receptor. Okay, so I hope you have clearly understood. Again, I am telling you, you have to write it down and practice. Okay, because especially G-protein coupled receptor, in the first go, yes, it is a bit problematic, but once you get it clearly, then you will not forget it anywhere. Okay, fine. Now, and in fact, you don't have to forget that. Okay, it's very, very much important in the whole pharmacology series. We'll be that time I won't be again explaining it. I'll be just telling you this is working on the basis of G-protein coupled receptor. So you should know what is G-protein coupled receptor. Okay, fine. Now here you have to understand about the comparative study. So if you compare the location of the receptors over here, the three receptors, if we talk about your ligand gated, we talk about G-protein coupled receptor, enzymatic receptor, all these are present in the membrane. All these are present in the cell membrane. But whereas your nuclear receptor, which is there, as I told you, it can be there in the nucleus or it can be there in the cytoplasm, right? So these receptors which are there, these are going to be in the intracellular space. This is going to be there in the intracellular space. Okay. Now the next one is going to be the effector. Okay. Effector is ligand gated. What are we targeting? We are targeting mainly the ion channels. If you talk about the G-protein coupled receptor, what are we targeting? The channels and mainly the enzyme, like the phospholipase enzyme, aryl cyclase enzyme. Yes or no? Fine. Now next over here, enzymatic receptor, name itself tells, the transmembrane linked enzyme. Okay. So what is that receptor linked enzyme? That's nothing but tyrosine kinase, which we were talking about. Then again, what are we targeting? What is the effector over here? That's nothing but the enzyme. And especially in the nuclear receptor, what are we targeting for? We are targeting for the genetic transcription. That particular complex is going to bind with that particular specific site of DNA, which is going to alter the gene transcription. I hope it is clear. Now next is the example. What are different examples? You can see all the examples over here. If we talk about the ligand gated. If we talk about the ligand gated channels or ligand gated receptors, you're having nicotinic, GABA A receptors. Then if we talk about G-protein coupled receptor, you're having muscarinic and adrenergic receptor. If we talk about enzymatic receptors over here, you're going to have the insulin epidermal growth factor. And then if we talk about nuclear receptor, as I told you, vitamin A, vitamin D, steroids and thyroid hormones. Okay. Now one more very important parameter to be looking under is the time taken for the action. What is the time required for the particular receptor to show the action? Okay. So for that, if we talk about ligand gated, it is going to take in milliseconds. If we talk about G-protein couples, it's going to take seconds. Then enzymatic is going to minutes to hour, and nuclear receptor will take comparatively more time, that is in hours. Okay, so I hope you have understood the lecture till here. Okay, this was all about the receptoral families, the comparative studies, G-protein coupled receptor. Again, I am telling, do refer our notes, compare with proper systematic way how you supposed to write the answer for 5 marks is given in our notes. Join our Telegram channel. Okay, keep supporting us. This is your lecture number 7. We will be coming with the lecture number 8, and soon we will be finishing this general pharmacology series. Thank you everyone for listening to me so patiently. All the best. Do well in exam. See you in the next class.